Yeah, I'm going to be talking about my PhD project today. And so to kind of design or to motivate the question, I am, and given the recent uptick in COVID, I'm going to take you back to a slightly, uh, yeah, just I feel like I had done this introduction at some point in the past. And I think maybe just given that it's uptaking again, it's worth using this again. Yeah. So in the early days of COVID, if you were looking for an N95 mask, for example, you would potentially have to find the two nearest pharmacy available to IMSC and estimate how likely you were to find these masks. For example, over time, you came to understand that you had an 80% chance of finding a mask at pharmacy 1, whereas you only had a 20% chance of finding a mask at pharmacy 2 whenever you went to each of these options. So my question or my project aims at understanding how we estimate these values that we associate with how likely we are to get a reward from a particular option, and how we would behave when having to decide between these two options. Um, Now, for example, let's say that over time, pharmacy 2 gets a new supply, whereas pharmacy 1 supply depletes, and these values change over time. How does the brain now update the value that it's associated with the two options and modify its decision-making process? So essentially, this kind of decision-making between probabilistic options in a rapidly changing environment is what I'm hoping to get some answers at. Where are the values represented? How are these values updated so that so that behavior arises? And so these questions have been asked, or versions of these questions have been asked in a bunch of different species, including humans, pigeons, and for this particular example, monkeys and mice. In the monkey task, for example, a monkey had to choose between two visual cues, a green cue and a red cue. And the red and green cues provided reward with different probabilities. The animal had to saccade or move a joystick to indicate its choice. And upon making a choice, it was rewarded with the probabilities that were associated with the two options. Similarly, in a mouse task, the mouse had to choose between a left and right lick port, each with different probabilities of producing water. And these were water-deprived mice, so they were motivated to perform this task. And what we see, what has been seen in these kinds of experiments is that, when in blue, you're plotting the ratio of choices that the animal makes, so the probability or the choice ratio of choosing green versus versus red, and similarly the income ratio, which is the ratio that the animal receives a reward for choosing green by the value of the number of times it receives a reward for choosing red. And you can see that these two curves overlap even as they change over time. And this behavior is called operant matching. So the ratio with which the animal divides its choices matches the ratio with which the animal receives an income. So in this example, in this block of trials, for example, the animal receives a reward from green much more often than it receives a reward from red. And it also chooses green equally more often than it chooses red. And similarly, mice also show this kind of matching behavior. If you average over blocks where the income ratio has remained constant, and you plot the choice ratio versus the income ratio, if there was a perfect matching, you would see that these points lie along the unity line. But what mostly is observed is that there is a little bit of undermatching, where as the ratio gets closer to being completely one or the other option being rewarded, the choices are a little bit more random. So there's a little bit additional exploration even when the animal receives a reward completely from one option. And this is thought to indicate the animal's willingness to explore and kind of see if the environment changes over time. And so to be able to do something like this, you need to have a representation of the value or the reward history that the animal has received from making a particular choice in the past. And that's how you can estimate how often you are, or how often you've got a reward from option 1, option 2, and then calculate the ratio. And so these kinds of value representations have been found in both the monkey and the mouse. In the monkey prefrontal cortex, for example, there are neurons that respond to a given visual cue, say the green cue of the green versus red, and the amount to which these neurons are active depends upon the objective value that has been assigned to that option. So if it has received more reward in the past, it's more active, and if it has received less reward in the past, it is less active.
And Adithya, so so there is a question in the chat.
Sure.
From Zita Bro. Is operant matching just probability estimation from relative frequencies? How is it about the previous slide?
So I guess in the example where so in the example that I provided, the two probabilities added up to 1. And so maybe I think you can see that a little bit more. So you can call it probability matching, where they basically just estimate the probabilities and choose that. But I think in situations where the trials don't sum up, the probabilities don't sum up to 1, or where they are like, say, 0.2 versus 0.1, it's the ratio that is important. So I guess in some ways it has the probability, but it's the probability, that's the relative probability of the two options. Yeah.
So Sitapro, does that answer the question?
Yeah, it's fine.
Yeah. Okay, please continue.
Yeah, and so these neurons, the activity of some of these neurons correlates with the object value for the two options as calculated by the experimenters. And similarly, in the mouse prefrontal cortex, there have been neurons whose activity represents the relative value or the value difference between the two options that the animal has to choose between. So in the bottom, you can see the spike rate, and in the top is the relative value over trial number. And so you have this examples of how value is represented in the brain and can allow you to kind of estimate the ratio of rewards that the animal has received from either option in the past. But as I pointed out in my example, these values can change over time as the environment around the animal changes. And so the next question that needs to be asked is how these values are updated and what are the constraints that you need to put in place and the rules that underlie how these values are updated. And these are questions that have not been tested or answered as clearly in the mouse and monkey systems. And so, however, there are mathematical papers that have drawn some constraints on what the rules should be that underlie these kinds of updating. And so, in a simple paper, or in a paper with a simple model, where you have two sensory neurons that represent the two different options that are available, two output neurons, and reward information passing into the circuit, and the weights of the neuron to sensory neuron to output neuron synapse can be modified over time. You can get this kind of a circuit to perform this kind of two option task, and you update the rules by updating your weights. You update the value by updating the weights between the two sensory neurons, and then you pick the output neuron that is more active, or you have some kind of behavior function that uses the output of the two options to decide what the behavior should be. And in this kind of a paper, Lowenstein and Sung showed that you need a learning rule that updates these weights according to the covariance between is a covariance-based learning rule that updates it with a covariance between the sensory neural activity and the reward activity. And so and they showed that when in a learning rule where this tends to zero, the covariance tends to zero at steady state, that is essentially what matching behavior is. That's the kind of rule that you need to produce matching behavior. And the kinds of covariance rules that they looked at were rules where one of the terms involved had were mean subtracted and had an average of 0 or an expectation of 0. And so I wanted to test if this kind of a covariance-based rule was in fact what underlies matching behavior. But to do this, I made use of, sorry, I missed a slide here. And this is just to show that I tried to, I recreated the simulations in this task. And when I simulated this kind of a circuit with a covariance-based learning rule as defined by the authors, you see that the results lie along the unity line and show matching. Whereas when you have a non-covariance-based learning rule, you don't see matching. You see significantly more undermatching than you observe even the animals perform. And so to test whether this kind of a covariance-based rule is what underlies matching behavior in animals, I turned to the fruit fly. And to do this, I first had to design a task where I could get flies to perform these kinds of two-option multiple choice experiments. And to do this, we designed a Y maze that in which I could insert different orders in different arms of the Y and could track the fly's location in real time. So the fly begins in an, depending on the fly's initial location, the arm that the fly is in is filled with clean air, and the other two arms are given two different odors. And the fly gets to choose between the two odors and make its way up one of the arms. And if it crosses a threshold defined here in as by a black line, it is rewarded. And we reward the flies by activating sugar sensory neurons using crimson, which is a inverted channel rhodopsin. And so we get, and once the choice is made, the new arm that, so suppose the animal shows odor A, that arm is now filled with clean air, and then the other two arms are randomly randomly provided with the two odors, and the fly gets to make its choice repeatedly. And the rewards that are associated with odor A and odor B change over time. But before we could get to replicating the matching task that I described with the monkeys and the mice, there were some basic questions that we needed to answer with the fly. First, if this new arena was sufficiently good to induce learning, or the fact that the flies could associate stimulus and reward in the Y arena, whether they could learn from probabilistic rewards that there were relationships between the stimulus and the reward, and whether they could maintain two associations at once. And so in a task where one of the options was rewarded 100% of the time, and the other option was not rewarded at all, we initially measured just naive choice preference and found that the flies tend to choose pretty randomly. This particular example, the fly has a slight preference for the yellow option compared to the green option, but its movement is also a little bit slower. The choices are not very consistent. But then immediately after, when we start the same fly, when we switch over to rewarding the green option, after a little bit of a lag, it starts to very consistently choose the green option, suggesting that flies can in fact associate stimulus and reward in this Y arena.
I have a question here.
Yeah, go ahead.
So those two orders are chosen to be kind of neutral to the fly, that neither of them is something the fly likes or dislikes.
Yes, so for at the temperature and the concentration that we've chosen, the odors are mostly neutral to the flies. There are some individual preferences, which is why we initially started looking at a small amount of naive choices as well. But overall, we realized that for the most part, there is no preference. And so we have slowly reduced the number of pre-reward trials that we look at.
And there is a question from Arnab Pal. How does it choose between the two hands? Is it just a coin toss or it depends on other parameters? I guess initially before it learns.
So initially, so we looked at this in a bunch of ways. We looked to see if, for example, they just followed the walls or they had a preference for turning left versus right or something like that. And for the most part, it looked like it was just a coin toss. It wasn't always like a non-biased coin. There was sometimes, like as you can see here, there seems to be a little bit of a preference for the yellow option. But for the most part, on average, if we look over multiple flies, when there are no rewards provided for these two orders at this concentration, it's more or less.
Arnab, do you have a follow-up?
Yeah. Thanks.
Okay, please continue.
Okay. Yeah, and so I just looked at a bunch of different flies. Rather than showing the entire trajectories, I'm just showing you which option was chosen. The red arrow indicates when the reward was begun to be provided. You can see more or less even random choices before. And once the reward is provided, you get a consistent preference for the rewarded green option. And now we then we look to see if rather than rewarding it 100% of the time, if I rewarded it 80% of the time or 40% of the time, when the other option was not rewarded, what were the differences? And essentially, you can see that there's the rate with which the animal chooses the correct option is a little bit slower for as the probability of reward gets lower. But even for a probability of 0.4, the animal chooses the correct or the rewarded option more than it would by chance, which is the dashed line over here. And just to, now that we know that they can learn and they can learn from probabilistic rewards, we wanted to check if they could maintain two associations at once. And so to do this, we added a third order to the mix. And so here, in interleaved trials, the fly had to choose between a green and a blue odor. The green odor was rewarded 80% of the time. And then on every other trial, it had to choose between a yellow and a blue odor, where the yellow odor was rewarded 40% of the time. And the outgroup odor was not rewarded at all. And what I'm showing you here is preference before the rewards were provided and preference when the reward was available. And for both the low probability and the high probability odor, there seems to be an increase, except for this one fly, suggesting that they can maintain two associations at once. And so with these basic steps out of the way, we could turn to an operant matching task. And so in this task, in this particular example, the fly had to first receive a ratio of 0.8 probability of getting reward of 80% from the green option and 20% from the yellow option for the first 80 trials. This switched to a 0.11 and a 0.89 reward probability in the second, and similarly switched over in the third. And I'm just plotting the number of times it shows the green versus the yellow odor on each of these over the entire course of the experiment. And you can see that similar to monkeys and mice, flies also tend to match. And the only difference being is that it looks like flies take a little bit longer to learn what the new ratio is. So there's a little bit of a lag, but once you get past the lag, the slopes of the choice and income ratios are pretty comparable. And when I plot the average choice ratio for a given block, which is 80 trials in my case, we see matching with a little slight amount of undermatching, as you can also see in the monkey and mice behavior. And so it looks like flies also perform or choose between these two options in a similar way to these mammalian model systems. And so what is important is in deciding a fly's immediate choice is the reward history that the animal has received over several trials in the past. And so to test this, we ran a logistic regression that incorporated information about reward from the past 15 trials, as well as the choice that the animal made in the past 15 trials. And we found that the rewards all the way 15 trials in the past. So on the x-axis is the number of trials in the past that the information is being calculated for. And this is the bottom panel is choice and the top panel is reward. And so up to 15 rewards in the past, there seems to be a non-zero regression coefficient, suggesting that rewards in the past do in fact play a role in predicting the animal's behavior, with more recent rewards being more important than later rewards. There does not seem to be much of an effect of choice, except for the most recent past choice in determining the choice that the animal makes on the next trial. And this also agrees with what has been seen by Bari et al. In their mouse work. And so we made use of the time constants that we could calculate from these kinds of regression analyses to build a Q-learning model to try and predict choice behavior in the fly. And so what I'm plotting here is an example fly's choice ratio in blue and the model's prediction of what the choice ratio would be. And you can see that there's a nice overlap with the model even predicting these unexpected dips, because this is a dip where the fly chose to choose the unrewarded option for several trials, even though green was the more higher probability option, possibly because it received a bunch of rewards randomly at the beginning. Right. And so what we've seen so far is that flies do show the same kind of matching behavior or operant matching that monkeys and mice do in a similar task, and that they make use of the same kind of information about reward history in determining their choice. But the questions that we were hoping to answer in this system that couldn't be answered in the mouse or the monkey were questions about the learning rules that underlie how these values are updated over time, and understand how these changes in blocks can be tracked. Oh, I'm sorry, I missed a slide here. This is just to show you that looking at the log likelihood, when you have a model which only makes use of the most recent reward and choice information, it does much worse than models that make use of multiple rewards in the past to predict behavior. But yes, so now going back to the question that we initially were interested in asking, which is what are the features of the learning rules that are important for determining how these values are updated over time. This simple example circuit that Lowenstein and Sung used to make their claim about covariance, or a covariance-based learning rule, is actually nicely mimicked by the neural circuitry that underlies olfactory learning in the fly, which is the mushroom body. So the mushroom body consists of Kenyan cells, which are the primary cells of the structure, and these cells respond to odors. So you can, in a sparse manner, so that you can actually decode odor identity by looking at Kenyan cell population activity. And they synapse onto the mushroom body output neurons, or M-bonds, which have been shown to determine turning behavior as well as upwind locomotion in these flies. And the dopamine neurons mediate the synaptic strength between the Kenyan cells and the mushroom body output neurons, and are thought to represent reward or punishment. And as you can see, this is a nice there's a nice analogy between the mushroom body circuit and the simple circuit that Lowenstein and Sung used in their paper. And what we currently think of as the learning rule that underlies how these weights are modified is a learning rule that looks at coincidence of activity between Kenyan cells and dopamine neurons. So when the Kenyan cells and dopamine neurons are both active, the weights are modified. And these weights are, it's typically thought to be a presynaptic mechanism. And so it's not that the entire M-bond is now going to always be less active when one Kenyan cell and one DAN are active. It's just that if a particular Kenyan cell and DAN are active, that particular Kenyan cell's activity is now less likely to excite the M-bond. And so and the reason that it's less is that this is a depression-based learning rule. I'm sorry, I forgot to mention that earlier. And so there is a nice correlation here between the kinds of learning rules that were in the Sung and Lowenstein paper, where the Kenyan cells could represent the neural activity and the dopamine neurons could represent the reward activity. But we know, for example, that the Kenyan cells don't have a non-zero mean, don't have a zero mean response to odors. They have a non-zero response to odors. And even with adaptation over time, these Kenyan cells still are activated by odors. So we thought we could rule out these first two potential examples and focus in on this R minus ER times N rule. And so the difference between a covariance-based rule as determined by Lowenstein and Sung and a non-covariance-based rule is this R term. So if the dopamine neurons represented just a reward, if they just represented whether reward was present or not, they would likely be a non-covariance-based rule that was taking place at the synapse. If the dopamine neurons represented something that accounted for the expectation of reward that from a particular option. So if the animal chooses option 1 and it expects a reward 80% of the time, when it actually receives a reward, it will be less, these neurons will be less active. And so these are the two things that we want to kind of, when we translate the Lowenstein and Sung covariance rule to the Kenyan cell, this is what we want to distinguish between, whether the dopamine neurons have like a error signal or whether they are just a pure reward signal. And this is something that a couple of groups have tried to look for in the past in experiments that very closely mimicked mammalian experiments, but were unable to find signatures of this. And so we were trying to see if in our slightly more novel behavioral experiment, we could find signatures of a reward minus expected reward signal in the dopamine neurons. And so just to emphasize what the difference is between these two rules, in a rule where you just have a reward, and we know that the synapses get depressed when dopamine and Kenyan cells are co-activated, you would never see any increases in your synaptic weight over time. As trials go by, you would see depression whenever the animal chose an option and was rewarded, and basically it stays the same when the animal chose the option and was not rewarded. Whereas when you have an R minus ER rule, you do see an increase whenever the animal chose an option and was not rewarded, depending on the expectation that the animal has for receiving reward. And so you will end up at two different steady states. You will, in the rule that where the dopamine neurons represent just reward, you would always end up at a saturated, completely depressed synapse, no matter what the probability of reward was. It would just, the time that you take to reach that saturated zero value would depend on the number of rewards that you receive. And so when you compare, and so when I modified the Lowenstein and Sung rule to incorporate synaptic depression as the learning rule, and then looked at I'm sorry, the labels don't seem to show up here. But on the left, I am plotting a simulation of a modified Lowenstein and Sung model with a depression learning rule where the dopamine neurons represented just an R signal. You see that they basically completely show no matching. They don't have a preference of one option or the other, independent of whether they receive a reward, the ratio with which they receive a reward. Whereas when you have a covariance-based rule, they show matching. And the fly data in the middle looks a lot more like the matching behavior than it does, looks a lot more like the R minus ER rule than the R rule. But this is not, this is average over a bunch of flies and is not really strong in its predictive capabilities. And so we wanted to actually see if we could build a model that could test on an individual fly basis which of these two options, an R minus expected R rule or R alone rule, was better at predicting fly behavior. And so just to recap what I've said about the mushroom body, we have two different groups of Kenyan cells that respond to the two different odors that have different weights onto a M-bond. And what we know of M-bond activity is that it modulates turning behavior. And so when, say, for example, the animal makes its way up the Y and hits the odor boundary between air and odor 1, it may choose to turn less if odor 1 is highly rewarding, and turn more if odor 1 is less rewarding. And so you can use turning behavior as an output of this circuit and try and predict turning behavior using the modified regression model. And so the two different kinds of learning rules that could be occurring at the synapse could be one where the dopamine neurons represent just a reward, or one where the dopamine neurons represent a reward minus expected reward kind of error signal. And so we tried to fit the output of this we tried to design a regression model that mimicked this mushroom body circuitry and the learning rules that we currently know exist at the mushroom body circuit, to try and see if we could figure out what was important in predicting behavior. And so we had essentially what the inputs to our system, the inputs to our model were the choice history, the reward history, and a product of choice and reward history. Or the same things when, rather than reward, we had a reward minus expected reward. And we could use these inputs to predict which were important in determining behavior and potentially discriminate between whether a reward was sufficient or a reward minus expected reward was better at predicting the fly's turning behavior. And at the same time, given that we expect that these learning rules were based on coincidence between Kenyan cell and dopamine activity, we had an inbuilt sanity check to see if the regression was working right. We always expected that it would be the D term that actually looks at the coincidence between odor and reward in determining whether the weight was changed or not. So there was an inbuilt control in some way. And so this is just an example where I simulated turning behavior using a learning rule that incorporated reward minus expected reward, and found that it does, the regression model can in fact very nicely fit or predict the behavior. So in orange here, you have the behavior of the of the simulator, or the behavior simulated by the model, and in blue you have the predicted outcome learned or by the regression model. And so when we try to fit these two different learning rules to actual fly data,
There's a question from SK here.
Sure.
What about P minus E of P, where P is punishment? Why is a flight choosing a reward and not avoiding a punishment?
So we haven't actually given any punishment to these flies. Sorry. So let me just quickly go back to the experiment design here. We always chose to reward. The only thing we were providing them was the reward. The reason we didn't go with punishment was that, so historically, the rewards that have been provided that are not real food have been dopamine neuron activation. And these, if you activate the dopamine neuron to either the dopamine neurons that respond to punishment or the dopamine neurons that respond to reward, you could bypass this error signal because you're just overriding anything and just causing the neurons to be active to the same level. And so we wanted to let the natural circuitry do its thing. And if it needed to calculate an error, it would calculate an error. We didn't have a clean driver that we could use to target sensory pain-inducing neurons, for example. So the only way to incorporate punishment would be to activate the dopamine neurons artificially. And the reason we didn't have, say, like a shock board or a food port is this was something we tried early on. But in the design of this Y maze, incorporating a real punishment or a real reward was just hard. And so all of this is with just a reward in mind. We haven't punished any of these flies.
Yeah, please continue. I mean, SK is asking noxious odor question mark. But yeah, that.
So we wanted.
I think you got it.
Yeah, so we wanted the odors, since we are trying to get the flies to learn the value of the odor, we didn't want the odor to be intrinsically valuable. So choosing a noxious odor would work, but we didn't want to have the odor to have too much of a value to modify. And so, right, and so it looked here from our from what we've seen that a regression or a learning rule that incorporates this error signal does better at predicting flight behavior. But that was just the first kind of test. And so this is just further clarity controls of what we saw from the model, where the terms associated with the initial weight, the choice alone, so the animal's stimulus history alone or the reward history alone had mostly a 0 regression coefficient. Bias was a little bit all over the place, and the term that incorporated the coincidence between reward and stimulus was the one that was consistently elevated and was important in predicting the turning behavior. And when we looked at the log likelihood or compared the log likelihoods between an R minus ER model and an R model, when you subtract the difference, you see a positive value suggesting that the R minus ER model is a better fit for the data. But apart from doing these kinds of modeling tests, the next couple of experiments that we have in mind were to see if we could actually use the optogenetic tools available in the Drosophila to bypass this kind of R minus ER signal. Since we have hints that there is an expected reward signal in the dopamine neurons, if, for example, we activated the dopamine neurons artificially as a reward, rather than activating the sensory neurons, we could potentially be overriding any calculation of an expected reward that's taking place. And so basically mimicking an R rule rather than an R minus ER rule. And this could, and so there are different expectations for what the output behavior should be in these cases. So if you look for matching, for example, you would see a significant amount of undermatching if we activated the dopamine neurons. This is an experiment that people are running in lab while I am here. So hopefully I will, once I get back, I will know what's happening. And the other potential option, based on the EM data set that has been generated at Janelia, where they mapped the more or less the whole brain using electron microscopy, we identified a GABAergic neuron that sends inhibitory input to the dopamine neurons that also receive reward information. And these GABAergic neurons, in fact, receive output or receive input from the mushroom body output neurons. So if you look at the circuit, given that it's the weights that seem to determine the value, when a Kenyan cell is active, the M-bond's activity should then also reflect the value that the weights represent. And so this could be the source of an error of an expected reward term. So whenever the Kenyan cells are activated by a given order, the M-bonds will sort of represent the value or the expected reward from that order. That would determine both the turning behavior as well as could potentially send an expected reward signal back to the dopamine neurons that conflict with the reward information that the dopamine neurons are receiving. And so another experiment that we have just set up the flies for that I will go back to test are ones where these GABAergic neurons are inactivated, constituently inactivated. And so we would be able to, again, change what could potentially be an expected reward signal to just a reward signal. And then the final thing that we're going to look at is imaging the activity of the dopamine neurons while we are training. So if we could compare how they respond when the reward is not predicted versus when the reward is predicted, this could provide insight into whether the dopamine neurons are actually modifying their activity based on expected reward. And at the same time, as I mentioned, the mushroom body output neurons are in our minds represent the value signal. And so even though we know, for example, that there are neurons that represent these kinds of values in the monkey and the mouse brain, looking at it would also be nice to have a verification that the mushroom body output neurons represent similar values in the fly brain, and could provide a nice, clean understanding of where all of these important information are in a simple circuit, and in fact, how they communicate with each other, providing a little bit more of a clear picture of how matching arises. And yeah, that's all I have. I'm excited to do some of these experiments once I get back. Just wanted to thank the lab, and particularly my theory collaborators, James and Ran, who are also group leaders at Janelia, who've helped a lot with the modeling effort, and John, Peter, and Tobias for helping design the Y maze. Thank you.
Okay. Thanks very much, Adithya. Any questions? Chandra has his hand up. Chandra, you can unmute and ask.
Hi, Adithya. So
also, you can turn on your video if you like.
Okay, where is that? Yeah. I suppose that's the correct order. Anyway, so in the case where you start off with basically having no reward whatsoever, and then you start the reward. I mean, you did the experiments where you had this 100% of the time, one of them is actually, providing some reward, and the other ones. What if you follow? If you follow that with the exact opposite, which is that the other direction has like 100% of the time reward, and the other one. How long does it take for the fly to, figure out, like, for it to sort of settle down and figure out, like, estimate the correct one? I mean,
yeah.
Is there some sort of hysteresis that it takes much longer once it has learned some direction as opposed to, let's say, starting afresh?
So this is something we have looked at this. I'm sorry, I didn't have it on my slides here. We haven't looked at it in that exact example where we switched from 100, 0 to 0, 100. But we've looked at how long it takes to reach a steady state from, say, when it goes from 80/20 to 20/80 or something like that. And that takes roughly 15 trials. And this is more or less independent of whether it's the first time it's learning something or when it switches. It takes between 10 and 15 trials per fly.
Okay, and some of that might be influenced by the fact that the way the reward works currently is probably is not so affected by fatigue and stuff, right? Because let's say but if you include things like, let's say you had a reward which, like for example, if it go back to the mouse example, initially to start off it will be more thirsty than after subsequent number of droplets. So I think things might change.
Absolutely. And so yeah, we had initially wanted to start with starved flies and give them food, but the food port that we tried to incorporate just didn't work. And so we have starved flies. We have starved flies, but we're using the opportunistic reward, so they don't actually, their hunger state remains more or less the same throughout, whereas the hunger decreases in the mouse example. And so you're right, it could affect learning. Yeah.
Okay. Thanks a lot. Nice job.
Yeah, Sita, bro.
Yeah, hi. So couple of questions. So one was, can you actually use this to figure out whether flies can detect periodic patterns? So, like what occurred to me when you were talking about this changing the choice ratio was, suppose you do it in a periodic fashion. So, like you go from, let's say, X is to Y to Y is to X in a, highly periodic manner. Let's say you use, I don't know, 5 trials at X is to Y, and the next 5 trials is Y is to X, and do this, let's say, for a few times. And then let's say you don't do it. Does the fly anticipate this, that in some sense, does it figure out that it's going to soon see a Y is to X, and does it, change the behavior only to realize that it made a mistake?
Oh, that's a really good question. I guess, like initially, since like I said, since it takes around 20 trials to reach a steady state, and we wanted a few trials once it reaches steady state to see if it reaches a fixed value, we'd need maybe 40 trials rather than 5. 5 may be a little quick. But I think it's something, given that we can push a single fly to do up to 250, 260 trials, we can definitely try it. But I think it would likely involve pulling the fly out and trying again on the next day and repeating this, which is again possible, but it just was outside of the scope of the initial experiment, so we haven't tried it. My guess would be that given everything we've seen, it would probably be able to learn the pattern, but I haven't done these experiments.
Okay, great. And my other question was actually more a general question. You mentioned mice, you mentioned monkeys, and basically Grosso is like, somewhat down the scale. So yeah. How far down the scale can we push? I mean, for example, would we expect to see this in ants? I know that, like in a colony of ants, they have done this kind of experiments where you have this Y maze kind of thing, but with a single ant, can you actually do this?
So I think you would have to motivate the task correctly. But given what I know about ant learning, I think it would probably be possible. Ants, bees are generally, at least to my mind, they seem smarter than flies. So I would guess that they would probably be doing the same thing.
Okay, great. Thanks.
Thanks. SK?
I had 2 things. That is, one is would it be possible to try this dopamine neuron mutant flies? That's been shown to be important in a variety of things. So if, for example, in your case, that would say there is no feedback at all, no? So what would you expect in that?
Yeah, so I guess in a dopamine mutant fly, I would just expect that they don't learn anything.
I mean, that could be checked, no? Whether there are any alternate functions, alternate ways, or does it just go randomly?
Yeah, so these flies, the version of these flies that we initially tried with just a, it was a reversed potassium channel to inactivate the cells. The channels didn't express super well, so we are in the process of designing a CRISPR knockout fly that should be ready in a couple of months. And so it's something that it's on my list of things to do.
Okay. Yeah. The other one was, is it the number of dopamine neurons that are firing that makes a difference, or is it the amount of molecules that are getting transmitted across the synapse that makes a difference?
That's an interesting thought. So there are, for the rewarding dopamine neurons that we are interested in looking at, there are around 60 cells. My hypothesis has always been that it's the amount of dopamine, but it's possible that it is also the amount of dopamine molecules. But I see the point that it could be in in this circuit that I have here. If there's some change in the synaptic strength here, that could just reduce the number of.
Yeah, it could be that not all dopamine molecules are on the other side wired the same way. No, so they don't. There can be multiple ways by which the dopamine could be on the other side. So that's a sort of a fallback option for the flies. No.
Yeah, absolutely. You're right. And so one thing that we are considering that could potentially distinguish between these two options is rather than when we do the dopamine imaging, rather than recording the calcium from the dopamine neurons, because we would only record from the axons and not the cell bodies. Yeah. If we were to record using a dopamine sensor that was expressed in the synapses of the Kenyan cells or the mushroom body output neurons, that would potentially distinguish whether it's the amount of calcium. If you had both experiments, you could potentially say what the difference is.
Yeah, sure. Okay, nice, thanks.
Thank you.
Thanks, SK. Any other questions? Anybody else? Okay. I have a question which is a bit bigger. So your learning rule is basically taken from this sort of neural network structure and sort of. Okay, so I guess I didn't fully understand it. So there's a more general question. So E.T. James kind of said, at least about humans, that human learning is essentially Bayesian, and his argument was that, in his view at least, Bayesian learning is optimal in some mathematically proven sense. So any organism that does not learn in a Bayesian manner will be selected against, and so evolution prefers Bayesian learning. That was his claim. So just out of curiosity, can you think of a Bayesian learning rule here where it's basically the probability that I will get a reward in part one, given the history of what I have seen so far? Would it make sense to try that kind of rule and see how it matches with what the fly does?
Yeah, so I think offhand, my math may not be great enough to make that transition, but based on what you've said, I think it sounds like sounds like a similar rule to this rule in the bottom, where we're looking at history and using the history to determine. So
it might work out similar. I'll give it a try.
It could potentially work out
similar. Yeah. Right. Okay, so thanks, Adithya, for this very interesting talk.